That's right. Accurate calculations lead to successful PCR. Let's imagine you are going to do your very first PCR reaction. It starts with understanding the research question you want to answer and then designing PCR primers. Subsequently, you order a PCR kit and do calculations. Let's begin with ordering a kit. Let's say a PCR champ on your floor told you to buy Promega kit, and that's what you did. In the PCR kit, you will find tubes or vials containing Taq polymerase enzyme, DNTP mix, which contains a mixture of DATP, DTTP, DGTP, and DCTP. Kit will also have Taq enzyme buffer, a bottle of nuclease-free water, and magnesium chloride. Of course, this kit won't come with the primers you want, as each researcher may use same PCR kit, but they might want to amplify different DNA sequence of their own interest. There are many vendors that sell oligos, that is PCR primers. IDT is one such. Let's say you log into IDT website and specify sequence of forward and reverse primers. You can either design your own primer sequences, which we will see in a separate tutorial. However, right now you are typing in primer sequences published by another research group. Let's say you order 250 nanomoles of each forward and reverse primers. Soon you will get an envelope containing dry powder of oligos, 250 nanomoles in each vial. This kit will also have literature which will tell you details of all components that are packaged. In this case, the kit has a total of 250 units of Taq enzyme, 2 vials of 1 ml each of 5x colorless buffer, 2 vials of 1 ml each 5x green colored buffer. You can use any of them, green or colorless. 1 vial of 1.2 ml magnesium chloride solution and 1 vial containing 200 microliters of PCR nucleotide mix. These are nothing but stock solutions, as we discussed in earlier videos. Company that supplied the kit may also suggest a standard PCR assay system. For each component, it suggests a range for its final concentration in the assay system, which is nothing but concentration of working solutions. To assemble the PCR assay system, you need to calculate how much of each stock solution you will aliquot and transfer into PCR tube and make up the entire volume to, let's say, 50 microliters. This way, each of the component will reach their working concentrations. Let's start with magnesium chloride to illustrate this point. Here, stock is 25 millimolar concentration, and let's say you want to use it at 1 millimolar in the PCR reaction. Thus, you need to dilute 25 millimolar to 1 millimolar in the final volume of 50 microliters. So let's use the formula C1V1 is equal to C2V2 as we saw in earlier videos, where C1 is the concentration of stock magnesium chloride, which is 25 millimolar in our case. V1 is the volume of stock magnesium chloride that you will eventually aliquot and add into the final PCR assay. C2 is the concentration of working magnesium chloride which is 1 millimolar, and V2 is the volume of working magnesium chloride, which is the final volume of PCR assay system, that is 50 microliters in this case. So C1V1 is equal to C2V2 becomes 25 millimolar into V1 is equal to 1 millimolar into 50 microliters. Taking 25 millimolar on the other side, which goes to denominator, we get V1 is equal to 1 millimolar into 50 microliters divided by 25 millimolar, and that yields us 2 microliters. Thus, we will take 2 microliters from company supplied 25 millimolar magnesium chloride solution and add it to an empty PCR tube. So should we add 48 microliters water to make up the volume to 50 microliters? This way, magnesium chloride will surely come to its final concentration of 1 millimolar. However, if we make up the volume right now and then add the rest of the reagents, volume of the assay system will far exceed 50 microliters, which will also change concentrations of all other components, including magnesium chloride. The right way to generate PCR assay system is to add 2 microliters of magnesium chloride and then calculate similarly for all other components and add them too in the PCR assay. Only in the end that you add water and make up the assay system to 50 microliters. This way, all the components of PCR assay come to their working concentration. So it's time to calculate for PCR buffer. PCR buffer is provided by the company at 5x concentration, whereas its working concentration is 1x. Let's use the formula C1V1 is equal to C2V2 to calculate the buffer amount to be added to PCR assay system. C1V1 is equal to C2V2, where C1 is the concentration of stock buffer, which is 5x in our case. V1 is the amount of stock buffer that you need to aliquot and add in the final PCR assay system. C2 is the concentration of working buffer, which is 1x in our case, and V2 is the volume of working buffer, which is the volume of final PCR assay system, which is 50 microliters in our case. So C1V1 is equal to C2V2 yields us 5x into V1 is equal to 1x into 50 microliters. Taking 5x on the other side of the equation, it goes into the denominator. We get 10 microliters. PCR buffer is provided by the company at 5x concentration, whereas its working concentration is 1x. Let's use the formula C1V1 is equal to C2V2 to calculate the buffer amount to be added in the PCR assay system. Here C1 is the concentration of stock buffer, which is 5x in our case. V1 is the volume of stock buffer that needs to be aliquoted and added in the final PCR assay system. C2 is the concentration of working buffer, which is 1x in our case, and V2 is the volume of working buffer. It is the final volume of PCR assay system, which is 50 microliters in our case. So C1V1 is equal to C2V2 yields us 5x into V1 is equal to 1x into 50 microliters. Taking 5x on the other side of the equation, we get V1 is equal to 1x into 50 microliters divided by 5x. This gives us 10 microliters. So we are going to add 2 microliters of magnesium chloride and 10 microliters of TAC buffer. Now we are going to calculate how much of nucleotide mix we need to add to the PCR assay. PCR nucleotide mix stock concentration is 10 millimolar, whereas final concentration is 200 micromolar. So we'll again use the formula C1V1 is equal to C2V2, where C1 is the concentration of stock nucleotide mix, which is 10 millimolar. V1 is the volume of stock nucleotide mix that you need to aliquot and add in the final PCR assay system. C2 is the concentration of working nucleotide mix, which is 200 micromolar in our case, and V2 is the volume of working nucleotide mix, which is the volume of PCR assay system, that is 50 microliters in our case. So C1V1 is equal to C2V2 yields us 10 millimolar into V1 is equal to 200 micromolar into 50 microliters. So for concentration here we have two different units, millimolar and micromolar. Since we know that 1 millimolar is equal to 1000 micromolar, so 10 millimolar is equal to 10,000 micromolar. So let's replace 10 millimolar by 10,000 micromolar, and we get 10,000 micromolar into V1 is equal to 200 micromolar into 50 microliters. So V1 is equal to 200 micromolar into 50 microliters divided by 10,000 micromolar. We'll cancel all the zeros, and this yields us V1 is equal to 1 microliter. Thus, we need to aliquot 1 microliter of PCR nucleotide mix to add to the reaction mixture. Now let's calculate the amount of upstream and downstream primers that we need to add to the reaction mixture. Well, if you remember, you ordered primers from IDT company's website. You ordered 250 nanomoles of each of the forward and reverse primers as DNA oligos, and you received them in powdered form. Now let's say you decide that the final concentration of forward and reverse primer in the reaction mixture should be 1 micromolar each. However, you got 250 nanomoles of primers. Now 250 nanomoles is absolute quantity which you got as powdered substance. However, primer that you need in the reaction mixture should be at 1 micromolar concentration, which is not absolute quantity but concentration in solution. So on one hand you have powder, on other hand you want to make a solution out of it. So first step is to convert 250 nanomoles of primer into solution by dissolving it in water. But how much of water? Let's see a simple logic. Final primer concentration in working solution will be 1 micromolar. Now if you dissolve your primer powder such that you make a concentration more than 1 micromolar, then you can simply dilute it and add it to the reaction mixture such that its concentration comes to 1 micromolar. However, if you dissolve your powder in large amount of water such that its concentration becomes less than 1 micromolar, then you cannot possibly concentrate it and make it to 1 micromolar. Now let's take an example here. We have 250 nanomoles of primer from IDT. To convert moles to molar, we need to dissolve it in water. 250 nanomoles, if dissolved in 1 liter of water, will give us 250 nanomolar. Since 1 liter of water is equal to 1000 ml of water, 250 nanomoles per 1000 ml will give us 250 nanomolar. Now let's convert this into micromolar. Since 1 nanomolar is equal to 1 by 1000 micromolar, if we divide 250 by 1000, we'll get that many micromolar. So 250 nanomolar is equal to 250 divided by 1000 micromolar, right? And that will give us 0.25 micromolars. Let's imagine we actually dissolve 250 nanomoles of forward or reverse primer powder in 1 liter of water, which will give us 0.25 micromolar of primer solution. If you remember, our working primer concentration in the reaction mixture is supposed to be 1 micromolar. Now if we make primer concentration to be 0.25 micromolar, that will be less concentrated than the working primer concentration, which is 1 micromolar, and we cannot concentrate it. Now let's imagine if we would have actually made a primer stock which was 50 micromolar concentrated. Now we could have easily diluted 50 micromolar primer solution to 1 micromolar primer solution. Now let's see how to make 50 micromolar primer solution from 250 nanomoles of primer powder. 250 nanomoles of primer can be diluted in large amount of water, let's say 1 liter or 1000 ml, or little amount of water, let's say 5 ml. When primer molecules will be added, they will spread out and make less dense 1 liter solution, while in 5 ml they will make very dense solution. Now, dense solution shows its concentration, which is number of molecules per amount of liquid. If you dissolve 250 nanomoles of primer molecules in large volume of water, you will make less concentrated solution. Whereas if you dissolve primer molecules in less amount of water, you will make highly concentrated solution. So 250 nanomoles in larger volume will be less concentrated. 250 nanomoles in smaller volume will make highly concentrated solution. So volume and concentration will have inversely proportional relationship here. So let's do calculations using ratio and proportion. 250 nanomoles dissolved in 1 liter or 1000 ml of water gives us 250 nanomolar or 0.25 nanomolar primer solution. 250 nanomoles in 1000 ml gives us 0.25 micromolar. So 250 nanomoles in how many ml of water will give us 50 micromolar. So let's do it by ratio and proportion. 250 nanomoles in 1000 ml gives us 0.25 micromolar solution. So 250 nanomoles in how many ml of water should give us 50 micromolar solution. However, so we would have multiplied the outer values and inner values. However, this is inversely proportional relationship. If you remember, we just saw that volume and concentration were inversely related. For inversely proportional relationship, last two numbers of the equation will switch places. Now we multiply outer number and inner numbers to get x ml into 50 micromolar is equal to 1000 ml into 0.25 micromolar. So this will give us 5 ml. Thus, if we dissolve 250 nanomoles of powdered primer in 5 ml of nucleus-free water, we will make 50 micromolar concentration of primer stock solution. This stock solution will be stored in -20 refrigerator, and it can be easily diluted to give 1 micromolar working concentration in the reaction mixture. So now we have a primer stock of 50 micromolar for forward as well as reverse primer, which we want to convert to 1 micromolar in the final reaction volume. So let's calculate how much we need to aliquot from the stock solution and add to 50 microliter assay system to make it 1 micromolar, and we'll do the calculations using formula C1V1 is equal to C2V2, where C1 is the concentration of stock primers, which is 50 micromolar in our case. V1 is the volume we need to aliquot from stock primers and add to the final reaction system. C2 is the concentration of working primer, which is 1 micromolar in our case, and V2 is the volume of working primer, which is the final volume of PCR assay system, which is 50 microliters. So C1V1 is equal to C2V2 will give us 50 micromolar into V1 is equal to 1 micromolar into 50 microliters. Thus V1 is equal to 1 micromolar into 50 microliters divided by 50 micromolars. We'll cancel the units. Cancel 50s. What we get is 1 microliter. So we'll add 1 microliters of forward and reverse primers so that we get the final concentration of primers as 1 micromolar in the PCR reaction system. Now let's calculate how much of enzyme Taq polymerase we need to add. Now Promega PCR Kit came with Taq enzyme at the concentration of 5 units per microliter, while its final concentration in the reaction mixture should be 1.25 units per 50 microliters. We need to find how much liquid we need to aliquot from the commercial enzyme preparation that should contain 1.25 units, and we will do it by ratio and proportion. So 5 units of enzyme in 1 microliter. So 1.25 units of enzyme will be in how many microliters? 5 units in 1 microliter. So 1.25 units in how many microliters? This will give us an equation. 5 units into x microliters is equal to 1.25 units into 1 microliter. So x microliters will become 1.25 units into 1 microliter divided by 5 units, and that gives us 0.25 microliters. So we will add 0.25 microliters of Taq DNA polymerase. Now let's calculate how much of template DNA solution we need to add. Now the assay system suggested by Promega tells us to use template DNA at less than 0.5 micrograms in a 50 microliter assay system. Now let's imagine you got your template DNA from bacterial culture. You extracted DNA using Pcr Kit, and upon spectrophotometry you found that you extracted 500 micrograms of DNA in all. And let's imagine that you diluted this 500 micrograms of DNA in, or dissolved it in 5 ml of nucleus free water. Now you decide to add 0.5 microgram of template DNA in 50 microliters of assay system. While you have a stock of 500 micrograms of DNA dissolved in 5 ml of water. Since 1 ml is equal to 1000 microliters of water, 5 ml will be equal to 5000 microliters of water. Thus, 500 micrograms of DNA dissolved in 5 ml or 5000 microliters of water will give us a stock concentration of 0.1 microgram per microliter of template DNA. So we want to add 0.5 micrograms of DNA in reaction mixture. We will use ratio and proportion to find the amount of liquid that we need to transfer from stock DNA into reaction mixture. 0.1 microgram of DNA in 1 microliter. So 0.5 micrograms of DNA in how many microliters? We will use ratio and proportion, and the equation we get from this is by multiplying the outer values and inner values. We get 0.1 microgram DNA into x microliter is equal to 0.5 micrograms of DNA into 1 microliter. So x microliter is equal to 0.5 micrograms of DNA into 1 microliter divided by 0.1 micrograms of DNA, and that gives us 5 microliters. That is, we need to aliquot 5 microliters of template DNA from the stock what you have and add it to the final reaction mixture to do a PCR. Okay, so now we need to simply make up the volume of reaction mixture to 50 microliters by adding water. In order to do that, all we need to do is add up all the volumes of all the other contents and subtract it from 50. This will give us 29.75. This is how you assemble single PCR reaction. However, typical experiments generally consists of 10 to 50 reaction tubes, including controls. It all depends on type of reaction experiment you want to set. However, you will probably never run a single PCR reaction tube. When you have multiple tubes, you cannot assemble assay systems one tube at a time, and you need to make what are called as master mixes. In subsequent video, we will see how to create master mixes and quickly assemble PCR reaction tubes and save time when you are dealing with experiments consisting large number of PCR reactions.